We eat food, and we eat it frequently, at least 3 times a day, if not more, once you start counting snacks. And what we eat no doubt has consequences for our health, our cardiovascular health, our risk for cancer, our energy level, and our body mass. So the food we consume impacts our health, and we consume it frequently, but how do we decide what to put on our fork? Well, the answer seems simple. We should eat what is good for us and not what is bad for us. We should eat broccoli and tomatoes and not lots of cookies. But what if broccoli tasted unbearably bitter to you? Even though you knew it was good for you, it would be difficult to enjoy eating it, and you would probably not eat it as frequently as other food. Or what if your previous medical history caused fatty foods to taste fantastic, making them almost irresistible? Even though you knew you should consume fewer cookies, it would be difficult to avoid eating just one more. And what if tomatoes started to become less tasty every generation? If tomatoes tasted like cardboard, they wouldn't be very appealing, and no matter how good they are for you, you probably wouldn't eat them. All of these examples point to the fact that what we put on our fork has to taste good to us. But what tastes good is a complex relationship between us and the food, between our taste preferences and the food's chemistry. No one has been more influential in studying why we like to eat what we do and how this affects our health than our next speaker, Dr. Linda Bartoshuk. Dr. Linda Bartoshuk is the Presidential Endowed Bushnell Professor of Community Dentistry and Behavioral Science at the University of Florida. She is an internationally known researcher specializing in the chemical senses of smell and taste, and serves as the Director of Human Research at the University Florida Center for Smell and Taste. Bartoshuk earned her undergraduate degree in psychology from Carleton College, right here in Minnesota. After completing a PhD at Brown University, she worked for the Natick Army Research Labs, where research related to food for military personnel was being conducted. Then she spent 35 years as a faculty member at Yale University. In 2005, she joined the faculty at the University of Florida. In addition to being an influential researcher, Bartoshuk has been a leader in her field. Last year, she served as the president for the Association for Psychological Science. Prior to that, she was president of the American Psychological Association Behavioral Neuroscience and Comparative Psychology Division and the Society for General Psychology, as well as the Association for Chemoreception Sciences and the Eastern Psychological Association. The brilliance of Bartoshuk's work has not gone unnoticed. In 1998, she received the ACHEM-S Award for Outstanding Achievement in the Chemical Senses. Bartoshuk has been elected to membership in the Society of Experimental Psychology, the American Academy of Arts and Sciences, and the National Academy of Sciences. And last year, she was featured in Science magazine. What you may not know about Professor Bartoshuk is that she actually started off as an astronomy major at Carleton College. Fortunately for us, rather than studying stars, she became one. It's my honor to present you Dr. Linda Bartoshuk, who will discuss variation in sensation and affect and how we all live in different taste worlds. Linda Bartoshuk.
Thank you very much. I want to thank Kyle and Christopher and Taylor for taking wonderful care of me. I am terrible with finding my way around, and I haven't gotten lost once while I've been here. I love being here. It does remind me of my days at Carleton, and I'm very much looking forward because I have a special request to make of you. I'm going to give you a questionnaire, and what this is a dry run for the next generation of questionnaires we're going to use in my lab. And I want to get some feedback from you. We'll come back to that, but I'm trying to give you first is sort of a roadmap for what I'm going to do here. The first thing we're going to do is do a questionnaire and some demonstrations. Then I'm going to tell you a little bit about, and by the way, we will find out if you're a supertaster or not. Then we're going to talk about supertasting and health, taste damage and health, But the final thing is something I added particularly because of the theme of this meeting. What about making healthy foods taste better? Can we do this? Is this a legitimate way to improve our health? Well, we're working on a project, the Tomato Project at the University of Florida. We have preliminary data I'm going to share with you. Okay, are you a Supertaster? Here we go. This is, first, before we do anything with the questionnaire, I have to give you some information about PROP. PROP is a medication that's used to suppress the thyroid in patients with Graves' disease. That's a hyperactive thyroid. You may remember that the first President Bush had Graves' disease, his wife had Graves' disease, and their dog had Graves' disease, which made me worry a little bit about the water supply in the White House. In any case, I never got to test President Bush, but he probably was treated with this medication, 6-N-propylthiouracil, PROP for short. He probably got about 200 milligrams a day, and this piece of paper that's in this envelope you've got has 1.6 milligrams. Now, the reason I'm telling you this so carefully is there is new information out about this. If you were taking this for hyperactive thyroid, and you took this dose every day for several weeks, you could potentially experience liver damage. When that surfaced, it worried all of us, and we wanted to make absolutely certain there was no danger associated with this. It's been looked at very carefully. Of course, logic would tell you 1.6 milligrams versus 200 milligrams a day for several weeks is probably not a fair comparison. And in any case, we now have a definitive decision by a pediatric endocrinologist who is reporting to NIH. There's no harm connected to this envelope. Nonetheless, if it worries you, don't taste it. If you have allergies that worry you, don't taste it. I'm going to show you what we can do, but I needed to tell you this before we got to the point of tasting it. What I'm going to ask you to do, everybody has the questionnaire? I'm going to ask you to begin by filling it out. And the top of it is demographic information. And I can't see too well because of the lighting, but if you have a question, just raise your hand and yell, and we'll try to deal with it. But we're going to begin by filling out the demographics, and then let me tell you what the next part is. Sensations come in different varieties. Philosophers divide our sensations into the 5 basic senses: vision, audition, taste, smell, touch. By the way, you notice we say there are 5, and in fact, if you count them up, there are a lot more. It's because Aristotle lumped too many things together into touch and claimed there were only 5 senses, and we've adopted his nomenclature. Anyway, there are those sensations like hearing, seeing, smelling, tasting, touching. Then there are internal sensations like feeling nauseated or thirsty or hungry or satiated. Then there are hedonic sensations, feeling pleasure from something, feeling displeasure from something. What I'm asking you to do is to rate many examples of these. You'll find them on the front and back of the sheet. And I want you to rate them in the following way. 100 represents the most intense sensation of any kind in any of these categories that you've ever experienced, and I want you to name that experience right under the 100. So your first task is decide what is 100 for you. Once you've decided, go ahead and rate all of these on both sides, saving taste test in the lower left of the second sheet, and we're going to go through that and do that together. On the back, where we have hedonic experiences, there are some that are known to be positive, like the greatest pleasure you've ever experienced is clearly positive. The greatest displeasure is clearly negative. But food hedonics are something else again, because you can like food or not like food. So you've got to attach a sign. If you like it, it's plus. If you don't like it, it's minus. Okay, everybody get started. Fill out the demographics. Fill out what 100 is for you, then start on the rest. Okay. Be sure in the middle. Okay. Great. How are we doing? Anyone having a problem with it? Has anybody finished page 1 yet? Okay, we got a few who have. If we don't all finish, what I'm going to do is go on and let you finish it up as we go, but we'll give you a few more minutes. Question? I can't hear you. Did you say, can you rate something you've never? Let me come over and listen. And if you find things that you think are confusing or wrong on this, write a note on the sheet. I want your feedback so we can make this better. How many people have finished 1 page? Okay, what we'll do is go on, let you finish that at your leisure, and we'll do the taste test part together now. So turn to the second side, and first there is a statement on the lower left that said, rate the sweetness of candy. Open your candy, but keep the wrapper, because you're going to save this candy. So put the candy in your mouth, taste it for sweetness. Rate the sweetness on the same scale, 0 to your 100. How sweet is this? And then take it out, save it in the wrapper for a few minutes. The Supertasters are going to want this candy in a minute. Has everybody tasted the candy? Open the little envelope and take out the round piece of paper. Now I have to tell you, I was once giving a lecture, and after I finished, someone came up and said, by the way, those people in the back row, throw their questionnaires away. They tasted the blue sticker instead of white paper. So I now remind everyone, now I'm going to show you what I can do. Do not do this. I'm a recessive for this characteristic. I carry 2 recessive alleles. I have no binding sites on my tongue for this compound. I put the paper in my mouth. I'm wetting it thoroughly in my saliva. I'm moving it around. Don't eat the paper, but you get through with it, take it out, put it back in the envelope. I tasted nothing. Go ahead and try it, but don't you put the whole paper in your mouth right away. Taste a corner first. If it's intensely bitter, rate the bitter and stop. If it isn't bitter, put more in your mouth until you can do what I did. Okay, how many of you could taste it? Put your hands up. Wow. How many thought this was just about the most bitter thing you've ever tasted? And by the way, if you feel that this bitterness went above the intensity you originally set at 100, give it an appropriate number above 100. How many didn't taste it at all, like me? Those of us who didn't taste it at all should be 25% of this room if this were a random sample. Those of you who thought it was horrendously bitter should be about 25% of the room. The other 50% would be in the middle. It's bitter, but it isn't too bad. Now I'm going to tell you what all this means, but what's particularly interesting is I will be tallying this up, and we will report back to you what the distribution was. Okay, now let me begin. If you have a spare minute, fill out the rest. But let me tell you some basic facts about taste and smell that make it easier for us to communicate the rest of this. First, taste has 4 qualities: sweet, salty, sour, bitter. Some of you may have heard of a 5th taste, umami. How many have heard of that? I am not a believer in umami, and if we have time in the question period, ask me about it and I'll tell you why not. We have 4 qualities in taste, but look at smell. We have many, many different qualities. And interestingly, in taste, you can use a taste word to express an emotion that has nothing to do with the sense of taste. I can say, she's very sweet, or he has a bitter outlook on life. Notice that I am borrowing the affect that comes from taste and using it as a metaphor in a sentence about something else. We can do that because taste has hardwired affect. You're born loving sweet and hating bitter, and we will come back to that. Smell does not. There may be some exceptions, but we haven't been able to find any yet. For the most part, smells are very concrete. The names of odors come from the object that emits it. That's a lemon smell, comes from a lemon. That's a smoky smell, comes from smoke. There is a very tight connection between molecular structure and olfactory quality, and that's important when we start looking at the function of taste and smell. But let me first tell you the one fact everybody knows about taste, and that's the tongue map. Sweet on the front, bitter on the back, salty and sour on the sides. How many of you have read about this in a textbook somewhere? I see a lot of hands. This is an example of the tongue map from a children's science magazine, 3-to-1 contact. And the tongue map is bogus. It's based on a mistranslation of a German thesis. The thesis was of Hennigs done in the lab of Wilhelm Wundt in Leipzig in 1901. And I used to be at Yale, so it gives me enormous pleasure to tell you that the mistranslation was done by Edwin Boring, a Harvard professor, and it is rumored that he didn't like women, so I'm even happier to tell you about it. Now, one of the most important facts about smell isn't widely known. There are actually 2 kinds of smell. There's orthonasal and retronasal. Now, orthonasal smell is smell going into the nostrils. It hits these bones, travels up here, and it goes up through a small slit up into the olfactory cortex. Retronasal olfaction is olfaction that occurs because you put something in your mouth that has an odor. And when you chew and swallow, that odor is forced up behind your palate, and then it goes to the same place as retronasal. Now that's interesting. If they go to the same place ultimately, are they basically the same thing? And the answer is no. It turns out that this retronasal olfaction, when it combines with taste, is what we call flavor. So when you say you taste chocolate, you're not tasting chocolate at all. You put chocolate in your mouth, you're perceiving a retronasal odor and sweetness in your mouth, maybe a little bit of bitter, and chocolate's entirely an odor. Now, flavor, retronasal olfaction, and orthonasal olfaction are treated very differently by the brain. Somehow, the brain knows which way that molecule came in. And let me tell you about a patient I had many years ago. She came to me, and I almost hesitate to tell you what had happened to her. She took one of these flip-top cans and licked the inside of it. You know how sharp they are. She slashed nerves in her tongue. So she probably had plenty of reason to come to me and complain. But what did she complain about? She couldn't taste her mother-in-law's lasagna. She described to me going into her mother-in-law's kitchen and smelling the lasagna, and it was wonderful. Notice, she's got retronasal olfaction. But when she put it in her mouth, it was like cardboard. I didn't believe her. I didn't tell her I didn't believe her. I'm very happy for that. But I thought, since she was suing the manufacturer of the can, good luck. that in fact, she wasn't telling me the truth, but she was. And it was years before I fully understood what happened. What happened is the brain has to have a cue to tell it whether the odor came in retronasally or orthonasally. And if it doesn't get the cue, it throws the information away. So she smelled the lasagna beautifully, but when she put it in her mouth, the normal taste, touch cue from her mouth was absent. So the brain threw the information away. And years later, I duplicated her experience. I did not slash nerves on a can. I anesthetized my mouth with a liquid anesthetic, and I ate a Hershey bar, and it wasn't chocolate. I had totally lost the ability to perceive flavor just by manipulating what was on my tongue. I didn't touch anything up here, and I should have been able to perceive the chocolate flavor, but I couldn't. We now have studied this. Derek Snyder, one of my PhD students, has just finished his dissertation on it, and there is no question that the brain treats retro and orthonasal olfaction as if they're completely different, and Dana Small, using fMRI, has even shown us that they go to different parts of the brain. So what do taste and smell do? Well, taste labels nutrients. It's that simple. Salty, we like salty as soon as we can taste it. The receptors are not mature at birth, but as soon as they are, we like it. Sweet identifies biologically useful sugars. We're born liking sweet. Sour identifies acids that can burn. This, we always wave our hands because some children like sour. We don't quite understand that. But strong sour, we all dislike. Bitter identifies poisons. We're born disliking bitter. Olfaction does not label nutrients at all. Notice I've listed the nutrients: fats, carbs, proteins, micronutrients, vitamins, minerals, and anti-nutrients, poisons. And we have taste is, fat has no taste. Carbohydrates are sweet, sugar is. Proteins have no taste. Vitamins are too dilute. Salty and bitter are the taste of minerals, and poisons are usually bitter. So taste does label certain nutrients. Now you might say, I said fats didn't have a taste. How could I possibly say that? Olive oil doesn't taste like canola oil. It's not taste, and it doesn't smell. You're actually perceiving impurities in the oil. The oil molecules are much too large to be tasted or smelled. Whenever you think you're tasting a fat, you're tasting something else dissolved in the fat, because fats basically have no smell at all and no taste. Proteins also have no taste. Again, you think that can't be true. Molecules are too large. You only taste fragments of proteins. So olfaction labels objects. It analyzes the molecular structure of an odorant and then puts a template of that odorant in our brain so we can learn to like or dislike the odor, because liking or disliking tells us what that odor did to us last time we encountered it. If it was accompanied by something wonderful like calories, you learn to like it. Any surprise, you like high-fat foods, your brain is wired that way. If a flavor is paired with something bad, the flavor becomes disgusting. Ever gotten sick on something and never wanted to touch that food again? Conditioned food aversion, survival mechanism. But we live in different taste worlds, and this is where I work. The tongues of supertasters look different from those of the rest of us. So remember those of you who thought this bitter was incredible? Imagine that 100 you picked. Imagine the bitter of this probe paper being 70 or more on that scale you created for yourself. How many would have judged it as above 70? Do we have any that would have said it was above 100? I'd like to see you guys after the talk. You are the extreme ones we love to study. So let's look at your tongue. We're going to put blue food coloring on your tongue. Why? Because fungiform papilla are the structures on the tongue that hold taste buds, and they don't pick up the blue dye. So you dye your tongue blue with blue food coloring, and pink circles begin to emerge, and those are the fungiform papilla, and they're really easy to count and to photograph. So here's my postdoc, Lori Lucchina. She's swabbing her tongue. And I must be honest, I lost the slide of Lori's tongue. This is Valerie's tongue. I know every tongue in the lab. Notice the little pink circles beginning to emerge. Those are the fungiform papilla. Now, Lori is back on a table. In the old days, we videotaped tongues through a Zeiss microscope because we had sometimes elderly or ill patients who couldn't hold their tongues still. And if you videotape them, you can play the tape back, stop where it's in focus, and make a montage. But now we've got a better camera. So let me show you an ordinary tongue. These dots are the fungiform papillae. Every one of those contains 6 taste buds. Now, you can't see taste buds. They're buried in the tissue. Think of the taste bud like an orange, and the segments of the orange are like the cells. The orange is buried in the tissue, and there is a pore that leads from the top of the orange up to the surface of the tongue, and the cells have little extensions, microvilli, that go up into that taste pore. So if you were to multiply the number of fungiform papillae you see by 6, you now have an estimate of the number of taste buds. We can actually measure, we can count those taste buds in living subjects by simply taking one of these fungiform papillae and magnifying it, and under high magnification, the taste pore stains blue, so we see little dots, blue dots. We can count them. Now, this is an ordinary person. I'm going to show you the tongue of a supertaster. Look a little different? You wonder why sometimes different members of a family don't have the same reaction to foods? Just to help you see this, I've actually drawn a template. Now, this is a 6-millimeter circle. We got the idea for this from a high school student doing a science fair project in the lab. And she heard us talk about how to do this and went off to a stationery store and bought those little reinforcements you put on notebook paper. 6-millimeter hole, used those as her templates. I thought that was absolutely brilliant. So we are a little fancier. We bought a paper punch, and we punch holes in paper and stick the paper on. But that's the template we use, and it's very easy to photograph and count fungiform papillae. Now, in fact, I've done that for you here. I've outlined them in red. This supertaster has about 60 fungiform papillae in that template area, and I have permission to tell you this is the tongue of Steve Wooding, who is a very famous geneticist in taste. And would you know it, he's one of the best supertasters we've ever found. This is a fairly ordinary person down here, and this person has 16 fungiform papillae. Now, you know I'm a non-taster, I demonstrated that. I married a non-taster. This is a Mendelian recessive. Those of you who know the genetics will know how delighted I am to tell you that both our children are non-tasters. My daughter has 5 fungiform papillae in this area. Actually, you could almost superimpose the tongues of my family. What do we know about supertasters? Well, first, we know that women are more likely than our men to be supertasters. We know that supertasting varies by race. Caucasians have the fewest supertasters. In the United States, about 10% of the population have more than 35 fungiform papillae in the template area. I'm going to take a guess that we may have a few more than that in this room. The number looked high to me when I saw the hands go up. Are taste sensations more intense to supertasters? Well, how do we know? Now, I'm going to show you an example of an experiment we did, and we did it with 2 methods, an old method that we don't recommend using, and a newer method that I will explain as we go along. The older method is the kind of method you see when you take a quiz in a magazine, 9-point scale. You really love something, it's a 9. If you don't like it, it's a 1. If you're in the middle, it's a 5. The problem is you can't compare sensations across people because we don't know what 9 means to different people. Well, we're going to try to solve that problem. We're going to take 2 groups of people. One group is going to have a lot of fungiform papillae. They're supertasters. We're going to take another group with very few fungiform papillae. Now, we're going to give both groups a Coke, and we're going to ask them to rate its sweetness from 0 to the sweetest they've ever experienced. This is the old way of doing it. And when we do that, both groups do the same thing. Think about it. If you had to rate the sweetness of a Coke from 0 to 100, where 100 was now defined to be the sweetest experience you'd ever had, you'd probably rate it about 2/3 of the way up. Does that really make any sense? That people with many, many more taste buds taste the same sweetness? Well, let's do a second experiment and find out. Let's put earphones on, everybody. And now, we're going to ask them to dial the loudness to match the sweetness of the Coke. So they can turn it way up, they can turn it way down, but we're asking them to match loudness to sweetness. That's called crossmodal matching. And that was proved to be beautiful in humans by S.S. Stevens at Harvard in the 1960s. It turns out we are very good at this. If you take several thousand people and have them match a particular brightness and a particular loudness, you will get very close to the same match for pretty much most of the people in the group. Okay, what do we get when we ask them to look at loudness? Well, Our supertasters crank the loudness up to 90 decibels, loudness of a train whistle. Our other subjects crank it down to the loudness of a dial tone, 80 decibels. Now, we know a lot about loudness. We know that every time you add 10 decibels, you double loudness. So this is telling us that our supertasters are perceiving twice the sweetness in the Coke as the others do. And we didn't know that when we asked them to judge it from 0 to the greatest sweet they'd ever had, because we didn't know the greatest sweet varied. We do now. We know that supertasters, in your lives, the greatest sweet you have ever tasted is at least twice as sweet as it is for people like me. That's a big effect, and now we can start to show effects on behavior that have impact on health. Fundamentally, supertasters are living in a neon taste world, and it's interesting, I didn't understand this for years. When I color code my taste solutions in the lab, I use pastel colors. It's not an accident. Okay, the data I'm going to show you were all collected with the newer methods where we use a standard unrelated to taste. And the way this works is, we know there's variability in hearing, but if we have enough supertasters and enough others, we have no reason to think the variation in hearing is any different between these two groups. So if we take the average sweetness versus hearing, that's how we get this big difference between supertasters and others. Well, long story short, virtually everything tastes more intense to supertasters. These are just correlations for sugar, for tonic water, which is quinine, for salt, for citric acid from lemons. And you see all the correlations are significant. What's fun is we get an effect we didn't initially expect. Jalapeno peppers contain about 100 parts per million capsaicin. That's the stuff that makes all peppers burn. So we're going to take pure capsaicin, and we're going to give people capsaicin by swabbing it on their tongue and have them rate the oral burn on our scale and plot it against bitterness of prop, just exactly what you tasted. Look at the correlation. Our supertasters are perceiving way more burn than the others. And you watch the Food Channel, those people who are eating peppers, the one who wins the contest, clearly somebody here. Anyway, by the way, everybody desensitizes to peppers after a while. We can talk more about that if you want. But it turns out that supertasters feel more oral burn, and they feel more oral pain if they get a tongue lesion, and this is medically interesting. If you have therapy for cancer and you have mucositis as a side effect, lesions on the tongue, if you're a supertaster, your lesions are going to hurt a lot more than if you're not. What about creaminess? It turns out we did an experiment with dairy products, and we've got supertasters and others over here, and notice that the creaminess of, in this case, heavy cream correlates with prop, which is how we did this. We could also have correlated it with density of fungiform papilla. Why? Well, the reason pain correlates with taste is every taste bud's got a basket of pain fibers around it. The reason creaminess correlates with supertasting is because every fungiform papilla has touch fibers in its core. So that touch, and when you have dairy products, when you have anything fatty in your mouth, think about what you call the sensation. Oily, creamy, viscous. These are touch sensations. And so touch, if it's more intense, you're going to perceive greater intensity from dairy products. In addition, there's a connection between supertasting and flavor. Now, we should have known this because the food and beverage industry has been using this for years. Suppose you make grape juice and you want it grapier. Well, grape is expensive. Add sugar. The addition of more taste intensifies retronasal olfaction in a central mechanism we don't understand very well. But certainly the food industry has managed to make a lot of profits based on this. And you wonder why sugar is in everything? One of the reasons it's there is to intensify flavor through this mechanism. But I already told you supertasters experience more taste, so they're also going to experience more flavor. They do. So supertasters not only get more taste, more oral burn, more oral touch, they get more flavor. They live in a neon food world, not just taste. And this is a factor of 2 or 3. So this means we've got people having very different sensory reactions to food. So what does it mean if you're a supertaster? Because we can now make comparisons across people with this method I've described, where we use a standard like sound, we can connect that variation to health. And here is some information from that world. A variation from food affects diet, which affects risk factors for disease, and the ones I'm going to tell you a bit about are cardiovascular disease, alcohol consumption, colon cancer, and head and neck cancer. Supertasters and cardiovascular disease. Supertasters experience more fat sensations. I've already mentioned that. What that does is it makes them dislike high-fat foods. They don't like high-fat foods as much as others. It's over the top. They become cloying. Their high-fat food intake goes down. And by the way, all of these results are published. And if you want the references, I'd be happy to send you the papers. Their weight is a bit lower, although the weight effect is small. Their blood profile is healthier. They actually have lower cholesterol. Supertasters have a lower risk of cardiovascular disease. Just one slide. This shows you the first time one of my students found a relationship like this. This is the bitterness of prop plotted against body mass index, significant correlation. Supertasters on roughly have a lower body mass index. We picked a very good group. These are older women, and the correlation holds true in that group. It's not as good for some other groups. What about alcohol? Okay, alcohol is bitter to supertasters and irritates the tongue. Not surprisingly, supertasters don't like alcohol as much. They don't drink as much alcohol. Do they have a lower risk of alcoholism? Well, we don't really know for sure because alcoholism is complicated. You drink for a lot of reasons. You don't just drink because you like the taste of alcohol. So the jury is not in completely on this one. But here's a picture. This is my postdoc Valerie Duffy's work. This is drinks per week. And these, by the way, are social drinkers, not alcoholics. Here we have supertasters, here we have others. And note that if you're not a supertaster, you may or may not drink for any number of reasons. But the closer you get to being a supertaster, the more likely you are to drink less. This has now been demonstrated in several studies. Supertasters and colon cancer. Well, one of the major risk factors for colon cancer is vegetable intake. You've probably been told, eat your veggies, get a lot of fiber in your diet. It'll reduce your risk of colon cancer. Well, vegetables, many of them, especially leafy green vegetables, taste bitter. They also have texture that some people don't like. So the preference for vegetables is lower in supertasters. The intake is lower. And we did a study at the VA in New Haven, Connecticut, on men who were getting routine colonoscopies, and we counted the polyps in the colon. Now, this is a risk factor for colon cancer, and there was a correlation between the number of polyps and the bitterness of the prop paper that you just tasted. The thinking is that if you are a supertaster, you don't like vegetables, you consume fewer, that puts you at risk for colon cancer, and that's revealed by more colon polyps. That's just showing you in graph form. What about head and neck cancer? Now this is interesting because head and neck cancer has very different risk factors. The risk factors for head and neck cancer are smoking and drinking. Okay. Who do you think smokes and drinks? Supertasters or the other group? Anybody want to guess? It's not supertasters because alcohol doesn't taste good to them and smoking irritates their mouths more. So we get less smoking and drinking in supertasters, meaning the non-tasters have the higher risk for head and neck cancer. Notice we've got a single gene, the one that controls the bitter receptor for this characteristic you tested. You have one form of the gene, it raises your risk for colon cancer. You have another form of the gene, it raises your risk for head and neck cancer. Now, I'm not saying these are huge risks. They are not. We can find them statistically, but they're not huge risks. So just because you're a supertaster doesn't mean you're going to get colon cancer, and just because you're not a supertaster doesn't mean you're going to get head and neck cancer. Oh yeah, I didn't like that slide. It's too hard to explain. Okay, another link between sensory variation and health. It's not just our genes that make things taste different to us. There's pathology, and boy, is pathology common in taste. The nerve that carries taste from the whole anterior tongue, the part you can stick out, that nerve crosses your cheek, goes through your ear, right behind the eardrum, passes through the middle ear, and then goes a long bony passage through the skull and gets to the base of the brain. There are a lot of different things that can happen to that nerve on that path. So there is a lot of pathology to taste. And the one we concentrate on is the ear infections. Well, here's a picture. We're looking at the eardrum from the inside now. This is the malleus bone. This is the chordotympany nerve crossing behind it. Right there, naked, able to be attacked by any pathogens associated with ear infections. In particular, viruses invade the nerve. Viruses particularly invade bitter fibers because they're small and unmyelinated. Now, isn't that interesting? Bitter is a poison detection system, one of the most important taste characteristics, and it's so vulnerable that a simple ear infection can damage it. I wouldn't have built the system this way. Maybe there's some reason it's this way, but I would have protected that nerve a little more. What happens when you have an ear infection? Well, I've already told you about some of the interactions. I'm not going to take a lot of time telling you about all of these, but the fact is there are connections in the brain between taste and the trigeminal system, which codes pain and touch. Now, I already told you about anatomical differences for pain and touch. These are interactions in the brain, and they're inhibitory. This means that normally, taste input is inhibiting pain in the brain. Now, why would this be true? Think about an animal in evolution, an animal in the wild, fighting for its survival with its teeth. The probability of, say, a tongue injury from a tooth, either biting its own tongue or getting bitten by another animal, is relatively high. Now, let's say you've been bitten and you have a lot of pain from that injury. Would you eat? Well, you might not. But if you start to eat, this mechanism of taste inhibiting oral pain will click in and it'll reduce your pain. In fact, the next time you have a headache or some pain source, get a candy and rub the candy on the tip of your tongue to produce a sweet taste and see if you don't notice a momentary dip in the pain. You're stimulating that mechanism. So we have this beautiful mechanism that evolved to keep us eating, even if we had an injury that injured in the mouth and caused pain. And probably some early data suggest it may act with pain anywhere in the body. So taste is a pain inhibitor. Now, it also inhibits sensations like fat, touch in the mouth. So what happens when you damage the nerve? Well, several things happen. First of all, how many of you have heard of burning mouth syndrome? This is a horrible pain disorder that occurs only rarely, but when it does, it produces terribly intense pain, particularly on the tip of the tongue, and there's no visible pathology. We believe burning mouth syndrome is actually a disorder produced by taste damage in supertasters. What happens is you damage taste, you lose its ability to inhibit pain and touch, and you get a pain phantom in the brain. Well, what happens with otitis media? We're damaging taste. It releases the inhibition normally on touch, and fats start to produce more intense sensations. Imagine creme brulee, double its beauty, double its allure. Can you imagine you might want to eat a little more of it? So what happens to people who have ear infections as children, slowly they begin to like high-fat foods more and more, and they gain weight. Let me show you that weight gain. We're looking here at people with no otitis media versus people with a history of otitis media, usually in childhood. And by the way, this is not just an earache or two. This is a moderate to severe history of otitis media. And look at the females. On here, with the open squares, we see underweight or mostly normal. And here, we see overweight or obese. And notice that the males in particular show a huge increase in the number of people who fall into the overweight or obese category. Females show it too, but the males, it's really dramatic. I didn't show you the slide with Supertasters because I would have had to draw a bar way up here. But the fact is, this effect gets more intense for male Supertasters. In one sample, we had about 40 male Supertasters who had histories of otitis media. 2 of them were not overweight or obese, to give you some kind of indication. This is not a small effect. One of the reasons it interests me is because it's environmental. We heard yesterday from Jeff Friedman that so much of obesity is genetic. There's not a lot you can do about it, and that's one of the reasons he tells us to have some compassion for people who suffer from this. But there are some contributors to weight that are environmental, and those we can do something about. What do we do about ear infections? Well, that's not my area, but we sure should try to have fewer of them. Now, let me just show you, we did an experiment in the lab, and we took a series of dairy products, low fat, whole milk, half and half, heavy cream plus oil, and this is how thick or creamy they felt, the red bars, to otitis media sufferers, and these were the controls. You notice there's a very big effect here. We could pull this out in a lab study with a very small n compared to the thousands we test with questionnaire studies. This is retronasal olfaction. Notice that it's not just fat that goes up. These are foods, one of them wasn't significant, but yogurt, milk chocolate, and ketchup all showed intensified flavor in people who had histories of otitis media. And this is done with a very large group of subjects, and we asked them for food preferences. And by the way, they were the same food preferences on your sheet on the right of the second page. We did factor analysis. We got a sweet group, we got a fat group, and you'll notice that the otitis media sufferers like these foods better than do the others. Most recently, and this is what I want to end up by telling you about, the fact that we've developed these psychophysical methods that do such a good job of comparing Supertasters to others, people who have clinical damage to controls, we now can tackle problems we couldn't tackle before. When I got to the University of Florida, I got to meet some people in horticulture. They're in IFAS, Institute for Food and Agricultural Studies at the University of Florida. And we found we had common interests, so we started producing, we had a flavor group that meets every couple months. Harry Klee, David Clark, Charlie Sims, me, and a bunch of other people as well who aren't as intimately involved in the tomato project. Here's Charlie in his lab. He has a setup called CompuSense, which is a commercially available setup. We're seeing the back end. On the other side are 10 computers that subjects can enter their data on. We can bring these in 10 at a time, collect data, and I can walk in at noon and walk out at the end of the day with an Excel file with data from 100 subjects. Pretty nice. Here's some of the things we've learned over in Charlie's setup. What we're going to do now is we're looking at liking, disliking, and here is liking. And this 100 means the most intense pleasure you've ever experienced. This is the worst experience you've ever experienced. And you know that top? Of course, we all think it's going to be sex. No. Sex comes in at 8%. What is the top of here is spending time with loved ones. I like that. Anyway, what you see is when you look at Supertasters, you ask them to rate their favorite food on this scale, they rate it very high. Ask them to rate their least favorite food, they rate it very low. People like me are more in the middle. So we now know that Supertasters have very extreme likes and dislikes for food. Do they have extreme likes and dislikes in other categories? That's what we're going to find out with this questionnaire that you're giving me preliminary data on. We're in Florida. Orange juice is important. So I ran orange juice and grapefruit juice. Actually, a student did, Jacqueline Calva, did a lovely job. Turns out that orange juice is liked better and better the closer you get to being a Supertaster. But grapefruit juice is disliked, particularly by Supertasters. Why? They're so sensitive to the bitter of grape juice. This is Harry Klee. He is one of the world's experts on tomato volatiles. He knows what volatiles are in a tomato. These are the aromas. David Clark is an expert on flavor volatiles, flours, sorry, not flavor, flour. And what we did is, thanks to IFAS, we grew 53 varieties of tomatoes. And we took all of these tomatoes and ran them through Charlie Sims' CompuSense. And we asked our subjects to tell us how much they liked these tomatoes. In the chem lab, the tomatoes were all evaluated for the concentration of volatile, the concentration of sugar, and the concentration of acid. So we know what's in every tomato. We know how much people like the tomato. And the final player in this project is a gentleman named Howard Moskowitz. I don't know if I put a slide in about that. Howard Moskowitz has a company that does market research, and he has for years created food products. He does it with mathematical modeling by testing a huge range of products, getting hedonic measures of them. He can use modeling to optimize the hedonics and predict what ought to be a better tasting product than anything in the sample he started with. The fact that this works, you can measure by looking at something like, there's a Prego extra chunky spaghetti sauce. That was created mathematically, and it's one of the first great successes in the food world, and you can read all about it in an article by Malcolm Gladwell in the New Yorker. I think I wrote that in here. No, I don't think I did. So now, what are we gonna do? We're gonna use Howard's math to predict the perfect tomato. But in the meantime, let me show you something else that nobody's ever seen before, because nobody's ever done a study like this before. First of all, glucose is in tomatoes. And the correlation between liking and concentration of glucose is very good. We knew that actually already. Believe it or not, the major driver for whether you like a tomato or not is how sweet it is. Now let's look at some volatiles. Here's one, ethyl vinyl ketone. I tried looking up what these smell like, and I gotta tell you that the descriptions were not very helpful, but here's what's important. Notice that this volatile, the more of it there is, the more people like the tomato. Here's one that's significant, but not so much. People like tomatoes better if they have more of this one. Here's one that doesn't matter, and here's one, it's actually negative. If you create all those scatter plots, you can begin to see how you begin to design a better tomato. And by the way, those cardboard tomatoes in the supermarket, you're probably all aware that they were bred for shipping characteristics, which tends to do a number on flavor, which is why we're now trying to understand how to breed them for flavor. So our intention is to find tomatoes that maximize all of these volatiles that are positively correlated, minimizes all of these negatively correlated. We don't know how many of these we're going to have to leave in to make it clearly a good tomato. But in theory, and I should say in practice, because we're doing it as we speak, we're going to create a liquid with the correct components to make it an extremely palatable tomato. We will have people sniff it and taste it and find out if, in fact, they agree with us. But I cheated. Notice this? That's really pretty impressive. But you notice I used a truncated part of the preference scale? Let us step back and look what happens to tomatoes. If you look at tomato liking in the context of all pleasure, this is greatest pleasure, this is greatest displeasure. You know, the trouble is the tomato never gets terribly high up there. Same data. This is, we asked people to tell us what the best tomato was they ever tasted and the worst. This is the question. Can we, by using this technology, make a tomato that will actually break through the tomato ceiling and give us a tomato that's actually better than any tomato you've ever tasted? We don't know, but that's what we're hoping to do. And if this approach works, believe me, it won't end with tomatoes. We'll be going on and trying it with other vegetables and fruits, with the idea that if we can take something we know to be healthy and optimize its flavor, optimize its palatability, will that contribute to a healthier diet? And I did have Moskowitz on here to thank him for the use of his optimizing mathematics. I should tell you, this mathematics, if you were a company and you want to develop a new product, it probably costs you $50,000 to get him to do it for you. He has donated this technology to us for free, and I thank him for it very much. Are we going to have a better tomato? Stay tuned. Thank you.
From the audience here, and that is Linda had promised to answer the question is, what is unami?
Okay, can you hear me? I'm on okay?
You're on.
Umami started out as an advertising gimmick. Glutamate in the 1940s was advertised. If you ever bought Accent, it used to be sold in a container that said "shake to taste at table," and the advertisements told you that glutamate would intensify the normal saltiness of your food. So if you shake it on your food to make it as salty as you like, you can save yourself eating sodium. There's only one small problem.
It's got some.
Glutamate is a sodium salt, but an inefficient one. It tastes less salty than sodium chloride with the same amount of sodium. So what they recommended to people was devastating. It would increase your sodium intake if you did that. Well, that successfully drove them into stopping the argument that the reason glutamate was so great in food was because it was a taste intensifier. So, and I actually know who did this. One of their employees came up with the idea of claiming it was a fifth basic taste because nobody could figure out what that meant, so they could get away with it and it would sound good and they would sell more glutamate. So then over the years, they funded a lot of research in different taste labs, very, very good taste investigators who were not part of anything corrupt at all. I mean, they were just interested in getting, a research grant, which was great. And lo and behold, glutamate receptors were found on the tongue by Nirupa Chaudhary, which is a very beautiful piece of basic research. But the fact that there are glutamate receptors in the mouth does not mean umami is a fifth basic taste. Here's what's really going on. We now know that there aren't taste receptors just in the mouth. They're all through the GI tract. You've got sweet receptors in your stomach. You've got bitter receptors in your stomach. You've got umami. You've got glutamate receptors and fatty acid receptors in your stomach. So consider, people claim glutamate is a protein marker. But how many proteins do you know that taste like glutamate? If glutamate is a protein marker, it's a bad one. But what really happens is protein molecules are too large to be tasted. So they're in your mouth, you don't taste them. You only taste other things mixed in with the protein, particularly with the fat. But you swallow the protein, and it's broken into its constituent amino acids in your stomach. Now you've got free glutamate in your stomach, and it binds to the glutamate receptors, and your brain gets the message, and it said, wow, this person just ate protein. I'm going to make whatever sensory cues came into the mouth before the protein liked better. It produces a conditioned food preference. So you learn to love roast beef and chicken and cheese and eggs, all the protein sources, and it was produced by conditioned preference induced by stomach. Now already, since these receptors in the stomach were discovered, the people advertising glutamate as a fifth basic taste are already hedging their bets, and they're beginning to talk about glutamate receptors in the stomach. So I think this whole history will just fade. It'll fade back. Nobody will pay any attention to it. And they'll begin talking about what really happens, which is actually much more interesting.
Okay, thanks. Questions or comments from our panel? Paul?
Well, I was already thinking about asking a question on the importance of a person's upbringing or culture, and now you've told us a little bit about conditioned food preferences, and I'm wondering if you could tell us a little bit about that and where we might see some differences in terms of the kind of foods that one gets used to eating. And
yes. I should say, because I work in taste, I am enchanted with anything that is related to taste. So I exaggerate about what the taste mechanisms are, essentially. The fact is, up to about 15 years ago, if you had asked me, I would have said virtually all food preference is learned. Mm-hmm. And it's all environmental. But then our psychophysical methods developed to the point where we could see this genetic variation, and we realized that for certain tastes, bitter and sweet, the genetic variation was really playing an important role. And so we began to concentrate on that and look at the risks for health. But by far, the more interesting, you might say, effect is the conditioned food preferences. And these occur everyday life. For example, this isn't everyday life, but cancer patients that are undergoing therapy are often made nauseated. Well, nausea is a beautiful way to produce a conditioned aversion. So what happens to our cancer patients? They develop aversions to their diet, which makes it hard for them to eat. Now, Eileen Bernstein did a brilliant study some years back showing that you could scapegoat that conditioning by giving a novel food just before you had the therapy. And in a group of children getting chemotherapy for leukemia, she fed them root beer flavored lifesavers. They wouldn't touch root beer after the therapy, but it preserved their diet at home. Talk about a brilliant and cost-effective behavioral intervention. It was really
the root
incredible.
beer people didn't like
They
it.
probably didn't.
I've got
Okay.
I've got
Marion?
one. I have a question. I thought your presentation was just fascinating. I've heard parts of it before, of course, with the blue tongue. But I'm really interested in condition preferences because one of the things that comes up about childhood obesity is that kids are conditioned, as I talked about in my talk, there's this business about kids' food.
Yes.
And appreciating the taste of kids' foods, and that kids don't learn to taste the wide complexity of flavors and textures of food. So do you think that that's going on as part of it, that these are conditioned taste preferences that are pretty immutable?
First of all, they are not immutable. You can extinguish conditioned preferences and aversions. You can form new ones. But I think you're absolutely right to worry about kid food, because one of the ways you can condition a preference is by watching somebody you admire consume something. Remember Mikey, that TV commercial? Mikey ate it. And so if you have pictures of cartoon characters, child movie stars, celebrities consuming kid food, it raises the value, and you can produce conditioned preferences that way. Probably could produce conditioned aversions too. I'm not sure anybody's really tried that. So I think you're absolutely right to be concerned.
Can I ask one follow-up question? There's the business about people's strong stated preferences for Coke and Pepsi, for example, and yet when you do blind trials, people can't tell the difference.
Well, actually, if you probably, they can tell the difference if you work it right. And my guess is what you're going to find out is that the Supertasters can tell the difference. And the people like me with a less exotic taste system cannot. And it probably depends on the mix of subjects you've got in your group. But I'd be willing to bet you that Supertasters can, because I know of Supertasters who can tell when Coca-Cola changes its sweetener.
Ah, yes.
Bina?
Yeah, you have this dichotomy between supertasters and undertasters. And I was wondering if it's possible for somebody to be a supertaster, say, at the bitter end of the spectrum and be an undertaster at another end, so that there's variation within the range.
Yeah. First of all, it's a continuum. And it's just for the sake of exposition that I make it look like a dichotomy, because it's much easier to design an experiment by using people at the ends of the distribution. But you hit on a very important point. We've got about 25 different bitter genes. Now, PROP, the one we tested with this paper, is only one of them. You can have variation for each of these genes. So it is conceivable. I didn't go into this because it's a lot of detail. But there are actually a few people in here who cannot taste PROP, but they have the anatomy of a supertaster. So they're a supertaster in every other way except for that one compound PROP. This may also be true of some other bitter compounds. In general, it's easier with sweet, salty, sour. If you are a supertaster for one, you're pretty much a supertaster for all.
Okay, thank you. Francis?
Thank you so much for a great presentation. I'm curious about the ear infection effect on desire for fat.
Yeah.
And did you tell us, maybe I missed it, the trend line for ear infection? Is there any correlation in terms of weight gain of our country?
You mean how many people have ear
Is there any-
infections and does it correlate with
Yeah,
weight gain?
yeah.
You know, I don't know. That isn't my field. I don't know how to do that well. If you do, I'll give you all the information you know to do it. I have no idea, but I would guess no, and here's why. I heard a talk about 30 years ago by David Levitsky, a psychologist at Cornell, that just changed my life. He pointed out that weight is normally distributed. Well, we know mathematically what produces a normal distribution. It's the summation of a large number of independent events. So the truth is, we know from the get-go that obesity isn't one thing. It's many, many things. And this ear infection is one thing. And I've been very careful to pick people who had the right history and so forth. Ear infections are fairly common, but how much of a contribution that would be to the whole country, I don't know. It certainly matters to individuals who've had a lot of ear infections. And I've had emails from people, you know, saying, oh, now at least I know why it is I can't resist barbecue ribs. Well, I can't resist barbecue ribs either. There are probably many reasons.
Okay, thank you. Question from the audience. What is your experience with the relationship between Sensatia and Supertasters?
What's Sensatia?
Oh, shoot. Synesthesia, sorry.
Oh, synesthesia.
Yeah, okay.
Yeah, absolutely. I don't know of any relationship, but it's never been tested. This would be something very interesting. If you have access to a popular, for those of you who don't know, synesthesia is an exquisite, I can't say abnormality, normal variant, where the senses, in the sense, if you hear a note, you can see a color. There are associations between, say, sound and light. Some of these associations involve taste. They're very, very rare. We actually don't know what the mechanism of synesthesia is, but Larry Marks has done a beautiful piece of work showing that those of us who use metaphor to compare senses are actually lining them up the way synesthetes do. For example, I'm going to tell you two colors, red and blue. I'm going to tell you two notes on a piano, high and low. Does red go better with high or low? How many say high? How many say low? The highs win. It is high. Red metaphor fits with the high note. And that's what synesthetes see. So it's almost as if synesthesia is an exaggeration of normal phenomena going on in the brain. I wish I had it. I think synesthesia sounds absolutely wonderful. Now, if you had a population, you could test them to see if they're supertasters and find out. And I don't know the answer, but that's a fabulous question.
Okay, great, thanks. Here's one from the internet. If we don't taste fat and only taste impurities in fat, can we make food taste good using artificial fatteners that aren't as bad for our health?
Yeah, you know, it's a great idea. Let's make fat substitutes. We know a dozen ways or more to simulate the sensory properties of fat. We can produce the mouthfeel, we can produce the taste, that is, impurities in the taste and so forth. But you know what happens after a while? The brain catches on. We are wired to want to take in fat because in terms of evolution, eating fat is a way to survive. Unfortunately, the things that make you survive on the short term when you're an infant are liking fat, liking sweet, and liking salt. Funny, they just happen to be the three major problems we run into with chronic disease later. Evolution doesn't care about you after you have your children. So we have all these brain mechanisms meant for you to survive. And now you put it, I'll give you the example that I love to use. Do you remember when McDonald's came out with a low-fat hamburger? First time I tried one of those, it was fabulous. Okay, you eat it, it was a marvel, and then slowly but surely it dies a painful death in the marketplace. Here's what I think your brain is doing. Your brain is saying, oh, interesting, hamburger, it's supposed to be fat. Where's the fat message? Something's wrong here, didn't get a fat message. I don't think this hamburger is good. It's a little too grainy, it does something else wrong. I don't like the color. And the next thing you know, you're giving yourself a lot of reasons why you don't like it. Your brain doesn't like it because it didn't come with fat. So as long as you try to have a low-fat product, you're also consuming high-fat versions of the product, high fat's gonna win. Here's what I think the food industry should try. Can you trick Mother Nature? Could you reduce the fat by 10% and not notice it and get a little healthier? Well, that's how I'd aim the research, but everybody wants a fat-free product. Here's our mistake. We should somehow educate people not to look for those cheap fixes, but to do the science, find out how the system works, and do what we can to jimmy it in our benefit.
Okay, thank you. How does super tasting correlate with ability to distinguish wines?
Oh, wow,
Oh, yes.
love that question.
Yeah.
I forgot to tell you, a few years ago, somebody asked me if chefs were more likely to be super tasters, and I said, no. I said, chefs, food's complex. A lot of things go into food. Then I got invited to a meeting with several hundred chefs, and I asked if I could probe test them, and they let me. There were way too many super tasters. So I'm thinking, why? I don't know. Wine tasting. I know there are people who've gone out and tried. I don't have their data. I'm afraid it's proprietary, and I haven't done that study myself. But wouldn't you bet there are going to be too many super tasters among wine experts? I'm going to bet on it based on the chef observation. But this is something where we need people who really know that world to go out there and do the studies correctly. And I don't know how to do that.
Okay, here's another one from the internet. Why is it that pregnant women's taste preferences can change?
Ah, love it. There's an interesting literature on the cravings and aversions of pregnancy. It's a very messy literature. It's not easy to make sense of it. But there are some things you can extract from it that look reasonable. For example, many of pregnancy cravings have to do with high energy foods, and there are higher energy demands during pregnancy. So this could be biologically wise. When you look at the aversions, one of the aversions that comes out on top is coffee. Amazing number of pregnant women don't like coffee. Well, consider, coffee is one of the most bitter things we consume voluntarily. The brain knows bitter is bad news. The bitter mechanism is hardwired to be essentially a poison detector. Is it possible that the brain keeps a list of all the foods you consume that are bitter, that it lets you get away with? But when you're pregnant, it says, I draw the line here. And now I can make the foods that you have that experience of bitter with not liked. Is that possible? I don't know. We actually don't have a good answer for this. Again, it's a wonderful question, and somebody ought to be doing research on that.
Okay, thank you. Another one from the audience. My sense of smell is 95% gone. How much does this affect my sense of taste?
Okay. Taste and smell, I would have told you a few years ago, are totally separate until we found the connection between taste and retronasal olfaction. So now we know there is a connection, and the problem is if you lose your sense of smell, 90%, actually, if the person who put that in would come and see me, I'll have a talk with you about what the cause might be. We know a fair amount about what causes loss of olfaction. But the truth is your taste for sweet, salty, sour, bitter will be intact unless the source of the loss was something that also gets taste, and viral infection is one of them. Viruses, upper respiratory infection, and flus take a toll on olfaction and taste. The mechanisms are different, but both senses are vulnerable.
Okay. I think I'm interpreting this one right. And, is there a link between high IQ and super tasters?
Funny you mentioned it in that direction. I've always thought if there was going to be anybody smarter, it was going to be the non-taster. I actually got a manuscript to review that made that claim a few years ago. It is not true, to the best of our knowledge. But let me tell you what is interesting. In the old probe literature, we find occasional papers relating personality characteristics to probe tasting. And they were pretty much dismissed by people like me until some very good people were able to replicate them with modern methods. And I think we've got to be open to this. I shouldn't say probe here, but whatever makes you an anatomic super taster, whatever that characteristic is that affects your tongue anatomy, could be having a lot of effects throughout the brain and body, and it could be connected to a lot of things. Again, one of the reasons I asked you to fill out this questionnaire is for the first time, we're looking for some of those associations. We will find them from this room. In fact, I can tell you with this N, we will find a connection between ear infections and body mass index.
Okay, great. One last question. Are people who like food plain often super tasters?
Yeah. I have to qualify that because remember, supertasters love what they love and hate what they hate, but their set of loved foods is smaller than for people like me. And they hate a lot of things, and bland things are protection. I have a couple friends who are extreme supertasters, and eating out is a real challenge because these people have spent a lifetime. And think about a supertaster child being born into a family. We don't know the genetics yet. It's probably some kind of recessive. So you could have a supertaster child popping up in a family of all medium tasters, let's say. And think of that kid having completely different experiences from everybody else, and you're constantly pushing things at him and saying, oh, you'll like this, and he doesn't. Supertasters at the extreme end tend to get very wary about things, and sometimes the easiest way to deal with things is to go bland.
Okay, thank you. Thanks to all of our panelists. We'll take a break now, and please join us back here at 3 o'clock. Please. Please remember to fill out your surveys, yes. They turned me on.